Thank you very much. Can you hear me? Okay.
Hello, Professor. Can you hear me?
Yes, I can hear you. Can you hear me?
Yes, I can hear you too.
Okay, I need to share my screen. Okay, thank you very much for inviting me to give this talk. I really appreciate the invitation. It's an honor to speak in front of such a distinguished audience. As mentioned previously, the talk is called Visualizing the Signal from the Noise. And my name is Robert Laramie. I'm from the School of Computer Science at the University of Nottingham. Since some of you may not have seen me or heard of me before, I thought I would just give a 30-second background on what I've been doing. So I was born in Boston, Massachusetts. My bachelor's degree is in physics from the University of Massachusetts at Amherst. My master's degree is from the University of New Hampshire in Durham, New Hampshire. That's in the US. And then that green arrow in the middle is when I moved across the Atlantic Ocean. So I wrote a PhD in computer science at the University of Vienna Technology in Austria. Then my next career step was as a lecturer and senior lecturer and associate professor at Swansea University in the UK. And currently I'm a professor at the University of Nottingham at the UK. So for anybody that's not familiar with the topic of data visualization, so data visualization is using computer graphics to generate images of large and complicated data sets. And that also includes large and complicated processes, for example, explainable AI. So the problems we typically work on are very large data sets, creating intuitive visual metaphors and designs for large data sets. We're always working on novel interaction techniques, trying to speed up the process of interaction. We're studying perception, how people perceive computer graphics and the images they see. We're looking into high dimensional data visualization, how to visualize and depict, for example, 5 to 10 or more dimensions. Of course, explainable AI and trustworthy AI are two of the very current topics today. We're looking at uncertainty, how to incorporate uncertainty into visualization. Of course, there are a myriad of applications in data visualization, and we're also looking at combinations of all of the above. So these are just an overview of some of the unsolved problems in data visualization, and usually the topics we're working on address some of these unsolved problems. Some of the research themes that we have worked on and up to now are things like visualizing computational fluid dynamics data, so CFD data, especially engine simulation data, so looking at how we optimize the combustible engines in cars, for example. We've looked at foam visualization, so this is actually computational physics, the study and behavior of foam as it interacts with surfaces. We've also studied computational biology, so molecular dynamics simulations. Molecular dynamics simulations result in very, very large data sets, so it's quite a challenge to visualize them. We've worked with marine biologists who generate lots of data using digital sensors. They attach digital sensors to wildlife, and then they try to understand what that wildlife how that wildlife behaved. We've studied sports and sports science, digital humanities. We've also worked on the more commercial side of data visualization, so visualization of call center data. We've worked on geospatial visualization and smart city data. And one of our current topics or major areas of focus is the visualization of health data. Another current focus we're working on is visualization literacy, trying to understand how people comprehend what they see. And then, of course, finance and visualization. And these are just some images that represent some of the topics we've studied and studied in the past and are studying currently: the smart city visualization, bioscience, business, understanding call centers and their agents, visual digital humanities, visualization of population health data, computational biology, high dimensional visualization, the CFD simulation data we mentioned previously, and data visualization literacy. Now for this talk is called Visualizing the Signal from the Noise. So I thought it would be interesting for this audience to look at one of the topics we studied, which was the visualization of animal sensor data. So one of the teams we worked with was with marine biologists, and marine biologists actually go out into the wild and they attach digital sensors to undersea wildlife. For example, leatherback turtles or imperial cormorants, albatrosses, penguins, and other wildlife. They physically catch these animals and attach very small digital sensors to these animals. They release the animal, and then the animal goes out into the wild for a few hours or a few days, and then they recapture the animal. They grab the sensor back from the animal, and they try to figure out what the animal did during that time that they were recording the sensor data. And the sensors record triaxial acceleration, pressure, temperature, and ultraviolet light, and amongst other different attributes of the environment. And then they recollect the sensors and spend weeks or months browsing through the time series data to try to figure out what the animal did during that time. So this is an example of a digital sensor here attached to a leatherback turtle. So the back of a leatherback turtle, and it records local acceleration. So that's how the body of the leatherback turtle accelerated as it's swimming, for example. It records local pressure, local temperature, local ultraviolet light. And then, like I said, the marine biologists actually recapture the animal a few hours or a few days later and try to figure out what the animal did during that time it was wearing the sensor. And this is what they're looking at. Typically, this is what the marine biologists are looking at. This is the triaxial accelerometer data. So the accelerometer data is recorded along three axes, right? The axis, the gravitational axis that points up and down, and then two other axes that can be considered as northwest orientation and then north-south orientation. And they claim, for example, that they can infer the behavior of some of these animals just by looking at this time series data, the three components of the acceleration data, and they say that they can infer some behavior just by looking at this time series data, which is quite difficult. Right? The red corresponds to what they call sway, the green corresponds to what they call heave, and the blue corresponds to what they call surge. And this is only 33 minutes of time series data, right? And then they go through and they try to read this signal and they try to infer, okay, this is what the animal was doing during this time. But as you can imagine, this is a very, very difficult exercise. So here's another example of trying to map out the behavior of an animal based on the time series data, looking at the 3 components of triaxial acceleration. In this case, somebody has chosen a time window indicated here and also indicated in the overview, and they're focusing on the behavior of the animal over the short period of time, those 10 to 20 seconds. And they claim that they can infer the animal behavior, at least some of what the animal is doing, by studying these time series data. But the question is, can we do better? Can we come up with some visualization metaphors that are better than the traditional time series data for studying the animal behavior? And that was our task during this project. The marine biologists asked us if we can do something better than this. So what we did was we did something that was sort of the opposite of what we learn in mathematics. In mathematics, when we're dealing with vector data, the mathematicians always tell us to decompose the vector into 3 components, right? If this is the original 3D acceleration vector, in mathematics we decompose the vector into the x, y, and z components. But actually, in this case scenario, the animal sensor data is already decomposed into the x, y, and z components, which is very, very unintuitive. So what we did was we reconstruct the vector back into its more natural and interpretable unit vector. So instead of deconstructing the acceleration vector into its 3 components, we take the 3 components and reconstruct the original vector. And then we position the origin of that unit vector at the center of the polar coordinate system as shown in this image. So now, instead of 3 separate components, we can look at the original acceleration vector in a polar coordinate system, which is much more intuitive than the 3 independent components. So in this image, we see the top, which is an overview of the original time series data in 3 different components. The user has selected a window of time to focus on, which is shown here. And then instead of the 3 independent components of the acceleration vector, we reconstruct them in the polar coordinate system. So they're no longer separated, but they form a unit vector at the center of the coordinate system, and then we can start to infer the orientation and behavior of the animal based on that acceleration vector. And what we see here, these yellow lines, that's the acceleration plotted on this spherical coordinate system over time. We're going to go into more detail about this as we go along. So this is what we call a better representation of acceleration. So this is what it looks like. Here we can see, for example, the acceleration vector in red, and we can actually attach a model of the animal to the acceleration vector and see the orientation of the animal over time. And I have a video here that should show that. So here is the acceleration vector, and it's showing the orientation of the animal over time, actually. And here's what the animal looks like. Here's what the acceleration vector looks like when the animal is descending through water, for example. because the animals start at the surface, they go through a descent, then they swim underwater, and then they go through an ascent. And this is what it looks like. So now we can actually see the orientation of the animal in a really intuitive way that's not really possible if we look at the three separate components of the acceleration vector. It's much more difficult to infer this kind of behavior just looking at the raw time series data. So once we reframe that acceleration vector back to the polar coordinate system, which corresponds to something in nature, right? Because we live in a 3D polar coordinate system, we can visualize it in different ways. So this is showing the acceleration visualized as a polar scatter plot or polar scatter plots. Right, the acceleration vector is, in this case, it's normalized, and that means every data sample is scaled so it fits on the surface of this sphere, and every point, every data sample is plotted on the sphere in the 3D polar coordinate system. This particular example is 9 hours of imperial corement data, and the activity is depicted as a polar scatter plot. So every data sample is plotted as a point on this sphere. And we can see just by looking at this 9 hours of data that clusters start to form already. So we can see some emerging patterns already from this data. So for example, here is a cluster already, and here is a cluster in these 9 hours, and so on. In this image on the right, this is the acceleration vector, but it's not normalized. So the magnitude of the acceleration is changing over time. And that's why some of these points are not on the surface of the sphere, because the magnitude increases over time and decreases over time. And we can choose to leave it unnormalized, the original magnitude, which results in a kind of point cloud like we see here on the right, or we can choose to normalize it so it adheres to the surface of the polar coordinate system. And we can also, so far up until now, we've only talked about the acceleration, but we can start to include other variates from the animal sensor into the imagery. So for example, we can include pressure. So this is the same 9 hours of imperial corement activity depicted as a polar line plot. So instead of the scatter plot, we've taken every data sample and we've connected every data sample by two edges, and those edges encode time, and they form a line. And then we can also incorporate pressure. So pressure is mapped to both the radius and the color. So if we start from the center of the polar coordinate system, this is the lowest pressure. And as we move away from the sphere, the pressure increases. So as the radius increases away from the center of the sphere, the pressure also increases. And that corresponds to the descent behavior as the imperial corment actually dives into the water and descends below the surface of the water. Because imperial corments swim underwater when they're foraging for food. And I forgot to mention this at the beginning, but one of the reasons that the marine biologists use these animal sensors is because the GPS doesn't work underwater. So they cannot use traditional GPS devices to study the animal behavior when they're diving underneath the surface. So they need to use these customized animal sensors that they call the daily diaries. And as we mentioned, the data samples are connected with edges for temporal information, And then we can see patterns. We can see the patterns of the animal diving, which is this descent underneath the surface. We can see these long edges, which represent the animal foraging or hunting for prey to eat. And then we see a bunch of lines that are zigzagging back and forth, and that represents the ascent back to the surface of the water. And all these blue lines represent swimming behavior at the surface of the water or above the surface. And we can also, now we can really infer animal behavior in a much more intuitive way. So if we look at a short time period of imperial cormorant behavior, we can see that there's activity at the surface of the water, that's swimming activity. Then there's a path of descent as the pressure increases. The imperial cormorant reaches a very, very consistent pressure and a consistent depth underneath the surface. And then it starts to swim in a search for food. And this is a kind of hunting behavior. The clustered acceleration at this higher pressure area. Then the swim continues, and then we see the ascent back to the surface, right, for oxygen. So suddenly that behavior that used to be or still is encoded in the 3 independent triaxial acceleration components can be represented in a much more visual and an intuitive way using the polar coordinate system and the multi-variate visualization. So it's no longer necessary to try to figure out what the cormorant did by looking at these time series graphs, which are very error prone. And we can also observe from this image, by the way, there's a different kind of swimming behavior during the ascent and the descent. So the descent is much more straightforward and moving in the direction of high pressure. The ascent is much less straightforward. In other words, one of the words we use to describe this curvature is tortuosity. So the tortuosity of the ascent is much greater than the tortuosity of the descent. And we're going to actually mention that in future slides. Another visual representation of this data we can create in order to study the behavior of this marine biology is popular orientations. So we can create histograms of the data, and then those histograms show the most popular orientations. In other words, the most popular orientations that the subjects spend most of their time in, right? In this case, the height of the bars is mapped to the number of data samples or acceleration data samples that are in this orientation in the polar coordinate system. So in this particular case, the tall or the long red bar represents the most time in which the animal spent in this orientation, right? And we can change these bins. The size of these bins is fairly arbitrary, so we can change their resolution with a user setting. So this is one resolution on the right, but this is a higher resolution on the bottom middle. But it's the same data, but just represented at 2 different resolutions. And again, as soon as we do this, we can see that clusters start to form. So patterns that were previously not so obvious start to become very obvious here, right? We can see clusters of orientation along this subtended angle of the sphere. And they're very, very distinct clusters that become quite obvious, actually. We can delve further into the clustering, so we can use clustering to abstract this data even further. So in the top left image here, this is what happens when we start to cluster the data samples of the acceleration based using the C-means clustering algorithm. So we can generate 1, 2, 3, 4, 5 clusters, for example, and then color code each cluster based on membership. And this is what the original sample data looks like. And using these clusters, we can abstract this behavior into a posture, what we call a posture transition graph. So we can represent each cluster as a sphere, and each sphere has its own color. And the transitions between clusters we can represent using edges. And as soon as we do that, we end up with a graph. So that is a graph, or what we call a posture transition graph. So the popular postures are represented by these clusters, and then the popular transitions are represented by the edges, and we can see that this is a very, very common pattern of postures and transitions between the postures. The transitions are not typically a straight line, so they're typically quite wavy, and we can represent that sort of waviness or any area that's not straight as a helix. And the helix is characterized by tortuosity. And tortuosity is a measure of how much a line deviates from the straight edge equivalent. So if we draw a straight edge from the starting point to the ending point, we end up with a straight edge. But that's not how nature behaves, right? An animal doesn't fly in a straight path, or it doesn't swim in a straight path. And the deviation from that straight path can be quantified, right? And it's a ratio of the length of the straight edge to the curved path, and that ratio is called tortuosity. And then in this bottom image, this is showing the combination of the posture transition graph with the actual data. And we can see the transitions, for example, in the actual data. And like we mentioned, we worked with marine biologists on this problem, and we also tried to get their reaction to these new visual representations of their data. So these are things, we generated images of their data that they had never seen before. And we got some feedback from them. So for example, they mentioned that the visual representations that we were generating were much more intuitive than their previous approaches. Figures 4b and 4d, these two figures, show the consistency of imperial cormorant behavior. So these are multiple dives. So this is showing multiple dives. This is showing multiple deep water swims. There's always this hunting behavior in the middle, and then there's always this ascent behavior at the end. And so this is a periodic behavior that's repeated over and over and over again. And one of the things that the marine biologists noticed is the difference between the descent and the ascent. So the descent has a much more kind of straightforward and narrow path downwards underneath the surface than the ascent. And the ascent has a very, very windy and kind of highly curved behavior before coming to the surface. And we believe this is on purpose or by design, so to speak, because the transition from the high pressure to the low pressure causes problems if the transition is too fast for the animal. The problems are described in using the term the bends, which is not a very technically correct term, but that's the sickness that an animal or a person can feel if they transition from a high pressure to a low pressure in too short of a time span. So it's a kind of prevention to prevent sickness due to the so-called bends. And this is something they were never able to see before. So they can study tortuosity in a more intuitive way and in more detail for the very first time using these representations. So those were all static images that I showed, but this system that we built is interactive. So what we're going to do is we're going to watch a short video that shows the interaction with this system, like how the user can interact and what it looks like when we're actually using the software. So this is the daily diary device. That's the animal tracking sensor. And here we can see the 9 hours of data being plotted in the polar coordinate system, and the user can rotate that view. So these are 3 views of the same data, and they're all connected, coordinated, and linked to each other. So any interaction in one view causes updates and changes in the other 2 views. So here we see the user focusing on a special window of time. The focus is shown in the focus view, and then the polar representation is shown in the polar coordinate system view. And in this case, the user has chosen to visualize both the acceleration data and the pressure, right? And now we can actually animate the samples over time, and we can actually watch the behavior of the animal as it descends into the high pressure or beneath the surface of the water, right? And the animal behavior comes out very, very clearly if we represent the data in this way. So all of the views can be animated, but we can always map time to animation. So here's the interaction with the spherical histograms that we mentioned, right? The height of the bars represents the amount of time or the number of data samples in that orientation or in that direction of acceleration. The user has control over the resolution of the bins. They have interactive control that's showing what happens when the user changes the resolution. And they also have control over the length, the start and end time, which they're looking at the data. And they can rotate the scene and view it from different angles. And then the patterns emerge, right? The popular transitions, the popular orientations. This is what it looks like when we start to cluster the data using C-means. We can assign a membership to each data sample. And in this case, the membership is fuzzy, so it's actually a probability. It's not a black and white membership. Every data sample has a certain probability that it belongs to a given cluster, right? And we can adjust that probability and that threshold to represent the different clusters. And then we can abstract the clusters away, and we can form the posture transition graph and add the edges between the transitions. We can indicate tortuosity. Transitions can be viewed by brushing the postures, and each transition can be colored according to its source posture. In other words, the orientation from which it came. So that was just showing. So we are running out of time. I'm not going to go into details about another follow-up project that we worked on. I will leave the paper references. All of these visualizations are described in papers that we can reference at the end and have been referenced during this in the slides. I'll just mention that we did work on other visual representations of this data, including a hybrid combination of parallel and spherical coordinates. So there are limitations to that work, and not all of the attributes are included in the visual representations there from the previous work. So we tried to add an experiment where we added parallel coordinates to the polar coordinate system in order to capture the remaining attributes of that sensor data. So the sensor data also has things like temperature. It has, well, we already saw pressure. It has infrared. It has an infrared light sensor as well. And so we tried experiments to capture that high dimensional data. I'll just show a little preview of what that looks like. I won't go into detail. This is the paper reference, by the way, if anybody's interested to see to read more about the details there. But here again, we have the three components of the acceleration vector that we mentioned. This is a window of time that the user has chosen to focus on. And here's that window of time in a zoomed in view. And then we can visualize all the attributes associated with that window of time on these parallel coordinates here. And then at some point we can connect the polar coordinate system to the parallel coordinate system and try to visualize all of the data at the same time in a high dimensional space, which turns out to be quite challenging. These are some of the experiments that we tried. So that's rotating the polar coordinate system while it's connected to the parallel coordinate system. And then you can see that there's overplotting, right? It's hard to see what's going on. This is the user adjusting the position of the polar coordinate system with respect to the other axes. And we tried some experiments to reduce the overplotting. So for example, by separating the sphere into two hemispheres and then plotting the data on this two hemispheres rather than on one sphere. And it does a little bit to alleviate some of the overplotting, but it's not full solution. It just shows some experiments we worked on. Okay, so I can see we're approaching 40 minutes. So I will wrap up now. I'd just like to give a quick thanks to some of the people that I worked with on this project. So Zhao Gang, Edward Grundy, Mark Jones, James Walker, and Rory Wilson. And I'm happy to take questions if anybody in the audience has any questions.